Yeah, so welcome to the last session of the day and before our dinner, of course, which we can use to talk about all kinds of things, scientific and non. I am in Bordeaux. This is Bordeaux. I think some people have actually been to Bordeaux from past Aptamers meetings. So I do primarily NMR spectroscopy and other techniques, biophysics, to look at how nucleic acids fold, how they interact with proteins when I was a bit younger, and now also some aptamers as well, and in collaboration with Philip, who you heard earlier today. Oh, this works great. Okay. So part of why we started to look at the atomic details of aptamers is that we all know that you can definitely select aptamers for very interesting targets. They can become very effective. They work, there's a little tweaking done to make them work better or in different conditions. But usually when you get to the structure part, it somehow stays at this stage in most papers. So I'm going to be talking about the dopamine aptamer, which started from this particular study here. But you can look at many different articles, and really it's all about optimizing. So you, in many cases, don't maybe even really need to know the actual details, but I hope that you will share with me the excitement that when you do know the details, a lot more information can be got, and then you can actually optimize and change how you're looking at it. And importantly, really see how is it that the aptamer is binding your targets. In this case, it's going to be small molecules I'm talking about. So that's also very interesting. How does a very small molecule be so specific for these aptamers? So I'm going to be talking about atomic details. You've already heard from Philip this morning about why NMR spectroscopy is quite useful. He was talking about it from an initial maybe screening standpoint. We can look at, for example, I'm going to use the cursor. For example, you can look at how an aptamer behaves when it's in the absence of a ligand. So in this case, this is a only very slightly modified aptamer similar to the dopamine one that was selected. And you can see that there's a large duplex region at the bottom, down here. And indeed, in the absence of ligand, you can see these signals here are indicative of a Watson-Crick normal duplex. And when you add the dopamine, you see the appearance of lots of new peaks, which when you heard from Philip, you'll know now means that there's some structuring, because we start to see a lot of the hydrogens that you can only see when something's in a base pair conformation. And one of the things when you're looking at it from a structure point of view from NMR, so for cryo-EM, you want to make things bigger so they're easier to see. And for NMR, often we want to make them smaller so they're easier to analyze. And that's one of the things we did here. We want to keep, of course, the binding mode, because that was the main question we were asking. So in a different series of experiments, maybe you can ask Rahit later about some of them. Yeah, the IT guy. Looked at certain aspects such as removing some of these duplex base pairs here to make it smaller, but maintain, as I said, all the other things that make it specific for dopamine. And this was great. And one of the other things about NMR, sometimes you are presented with these nice pictures. This is maybe not so big here, but there's lots of peaks. Each one represents a hydrogen. We can see this is the region where it's involved in base pairs, but you have to determine which ones they are. Like there's a lot of experiments to do this. And in order to not make any mistakes and to be unambiguous, and you can see why that's useful in the end, we took an assignment approach where we made many different oligonucleotides where only one of them was isotopically enriched for nitrogen 15 and carbon 13, which is the isotopes which are very useful for NMR. Which means we didn't have to guess at the assignment. We actually just made lots of different oligonucleotides. There'd be a single signal from each one. So for example, when it was just labeled on G9, there's only one signal, so we knew which resonance that was. And we did it not just for these base pair hydrogens, but for all of the sugar and base carbons, hydrogens, nitrogen. So it was unambiguous, which is actually very useful. It means that there's no mistakes made during the initial steps. And the other thing at this stage is that we told you a bit about looking at the hydrogens in the base pair, the amino ones, but the amino ones are also sometimes visible. And in this case, already at the start, we saw that in the complex, so the DNA, the dopamine binding aptamer bound to dopamine, you already start to see a lot of the visible amino hydrogens, which means that they're involved somehow in hydrogen bonding. So already indicating that it's quite an interesting structure going on. I show it for guanine here, but we also had similar results for if the T was labeled A or C. Again, just looking at these normal hydrogens involved in hydrogen bonding. So you can see that really there's a lot of hydrogen bonding. So it's probably somewhat complex. And I won't really go into too many details here, but through a happy coincidence, we found similar to the, oh, I don't remember which presentation it was now, that there was some inadvertent truncation, but this one actually bound better than the original aptamer. One of the talks just recently, that's what it was. So we took advantage of that because it was slightly smaller. As I mentioned before, smaller is easier, and it actually had higher affinity, and higher affinity is also very useful. So the particular structure we were looking at was it's very slightly smaller. So the duplex was removed and 3 additional bases. And this is what we used to look at the atomic detail to figure out really how is it folding and how is it interacting with dopamine. And I'm going to jump right to the structure part here. This is the same sequence that I just showed. And there's obviously some sort of folding. I don't know if anyone can see it right away. This looks a little bit like a duplex with a stem loop. But no, I hope if you see this picture now, you realize that it's much more convoluted. So instead of having the duplex, in fact, the 5' end, which is here, or maybe I'll use this for the online. The 5' end, which is here, and the 3' end, which is down here, are actually not together. So they don't come from a single duplex, as you might have guessed from the original M-fold prediction. Instead, you have regions that are somewhat 3 bases together. There's regions where there's maybe 4, and there's just a lot going on. So I want to go into a little bit more detail because I think a lot of the time, because of all these M-fold predictions, they are heavily skewed towards stem loop. Sometimes it's correct, at least at the bottom, because that was how they were, the capture selects, for example. It's probably going to be a duplex 5', 3', but then we often remove that. So through these elaborate selection processes, you can get anything that satisfies some sort of nice structure. And I want to show you that they can be quite diverse. So these numbers here that are shown in the structure on the left, if you were looking at an RNA structure, tRNA, ribosome, you would classify the base pairs, not just, oh, it's G, C, or A and U for the ribosome. You'd actually classify them by exactly what type of interaction is going on with the base pairs. And Watson-Crick is one of them, but there are many other ones as well. So that's what these numbers come from. This is from, you do RNA folds, there's different classifications. This is one of the ones, the Sanger one, probably, but there's other ways of categorizing them as well. The type of bases that come together, whether they are parallel or anti-parallel, which face are interacting with each other. So for Watson-Crick, there's only 2. And if you look at the structure here, there is only 2, and they're actually not beside each other. So we have one Watson-Crick up here and one down there. So already, this is hopefully you see that's already a little bit different. We only have 2 Watson-Crick, they're not together. This is unusual. If I go into a little deeper, I want to show you again some of the different characteristics. So now we have at the very top of this DNA aptamer, we have this interaction going on here. So here is one of the 2 Watson-Crick right here, C24 and G29. And you can tell it's Watson-Crick because it's 3 hydrogen bonds between G and C, and they are anti-parallel. The plus and minus is whether or not it comes out of the screen or into the screen, the phosphate backbone. But you can also see that there's both parallel and anti-parallel GG bases going on. And if I go a bit further, this is a GC, but you notice that it's a parallel. So this is a reverse Watson-Crick. And go a little bit further, we have TA. Again, TNA, not unusual, but these ones are also parallel. So yeah, reverse Watson-Crick again. You saw that there was the binding site of the dopamine. I'll come back to that. Here it almost looks nice if you were to look not so carefully. It's a part of the molecule that seems like it has a nice base paired region, a nice stack of them. But except for the top one, which is the second of the Watson-Crick bases, you can see that they look fairly good. In fact, here's a TA, but again, it's parallel. It's not anti-parallel. So you have all these reverse bases going on. So already that's different than expected. This is my favorite part of the molecule, I think. At the back here, you have what looks like they could be very nice base triples, but actually in none of the bases on this final strand here are planar with the two other ones nearby. So if you look here, it's a very zipper-like effect. And it's really nice, in my point of view, very satisfying that all of the hydrogens are hydrogen bound to nitrogens nearby and oxygen. So it's fully satisfying the different hydrogen bonds that you can make, but it's staggered as it goes down. So I think already this is, at least to me, it was an important lesson that because there's selection, I think you can really access quite unusual structures of DNA. And hopefully you see that already from this example. When we did the structure, I showed you the structure derived from the NMR data. Very often we also see what it would look like if you remove the restraints, the different distances that we put into the structure. So this here shows molecular dynamics of a microsecond with and without different the NMR derived restraints. And what we do is we tend to look at them in many different aspects. For example, you could look at the sugar pucker in this case. And if you look in detail, you'll see that almost all of them have the normal DNA sugar pucker. So despite the fact that this is quite unusual, it's actually very usual when you look at base by base. And you see there's very little dynamics in a broad sense. So this T23 is this one here. And it is dynamic by NMR. And you can also see in simulations, it goes to between at least two different states. Also in the category of being completely normal is almost every base is in the anti-conformation, which is again normal. There's only one that's not. You can see here, G31, which is just on this quartet plane up here. But everything else is in quotes normal. So despite the rather convoluted shape, nothing unusual really is going on from base to base. And kind of as a final way to looking at this ensemble, if you look at the appearance of let's say, a classic hydrogen bond geometry and distance over that microsecond, you can see that if we have the NMR restraints, it's in the first column. These are all different hydrogen bonds. You don't have to read it, but just each one is a different hydrogen bond in a base pair. If you take away all the NMR restraints, it's still mostly the same structure. By eye, it's identical. There's some different breathing motions, but it's still very similar. I also include a final category here. We took the structure, we took the dopamine, removed it, but kept the rest of the structure as it was. And you can see here that parts of it remain intact and parts of it are rapidly lost. And it's probably no surprise that the bottom is maintained actually, but this loop here that only forms when the dopamine's there. So as soon as you lose the dopamine, there's a broad opening of this cavity. But I think that movement's actually quite interesting because I think some of these aptamers, they take a lot to fold in the first place with the ligand, but after they're formed, you can lose and regain the ligand much easier. And I know ochratoxin was like this. We had this strange situation, but even 10% of ochratoxin would more or less fold all of the ochratoxin aptamers. So there's some interesting dynamics to be further probed as well. And finally, for this aptamer, looking at the details, we have the dopamine binding itself. So the dopamine's in yellow. It's bound by a nice little page of properties. So if we look in more details, the top one is a link plot type representation, and the bottom is just using pi mole or another sort of technique to look at the structure. And in this case, this highlights the part that is solvent exposed from the dopamine bound to the DNA aptamer. And I'm going to keep those there to talk about some of the binding properties. So dopamine is shown in the middle here. And if you compare dopamine to some of these analogs, all we did is add or subtract small groups. In the first case, there is just the addition of a hydroxyl group. Oh, okay. Hello. I think I moved the cursor off once again. Yeah. So by adding the hydroxyl group, you can see that it's in an area that's completely solvent exposed. So it has actually no effect. So the normal dopamine is 0.8 micromolar. This one is 0.7, so no effect. Makes sense. We can add it to the aliphatic part of the molecule. Again, it's solvent exposed down there. And again, it's fairly consistent. Perhaps it's a little bit closer to the surface or changes some dynamics of that part of the dopamine. But when you start to get more dramatic, for example, the this oxygen back here is definitely involved in hydrogen bonding to T18, as you can see. But also it is within the central cavity of the binding site. So this has a much more dramatic effect. You go from 0.8 to 100. So that's already going up by quite a large amount. And it becomes even more extreme when you put it on the back oxygen, which is involved in, well, 2 hydrogen bonds, but it's also even more buried in the capsule behind in the cavity, and it's now changing by 1,000-fold the affinity. So at least it makes sense with all the structural information. However, if you remove parts, it's also quite important. If you take away that hydroxyl, the same one I was talking about, it now drops to a 100-fold effect. So removing the hydrogen bond potential clearly has an effect as well. If you take away the aliphatic part, it doesn't bind at all. So it may look like it's a region that's not so important, but clearly it needs this bridging effect of the ionic interaction as well as this hydrophobic and hydrogen bound aspect. If we remove slightly the length of the amine, pulling it a little bit away from the bases and the phosphate backbone, it has a 10-fold effect almost. But if you swap that now, so it's no longer positively charged, but negatively charged, it again doesn't bind at all. So all of this makes complete sense. And so now we know why it's selective, but we also know why it overall, how does it look? And this last bit of information was really important. And I like it because it shows that not only is it informative and very illustrative just how complex DNA can be, but also helps in design as well. So this is used with the same Philippe that Philip talked about. And the standard approach, in this case, it makes sense. You have the attachment to an electrode at the 5' or 3', and then at the other end, you have, in this case, methylene blue to make these electronic aptamer-based sensors. And that's great, but in this case, neither of those had a very good signal. The first one, the one in gray, had no signal at all, and the second one had a very weak one in the inverse direction, so it must be pushing it further away. But if you look at the structure itself, already you can see that actually in this loop here is for sure the closest part of the folded aptamer to the electrode. And we know from the structure in the NMR that the base that's here doesn't actually form any base pairs, so it's free to be modified. So if you modify this one instead, what you can see is it now makes a nice, useful biosensor yet again. So it's not something you would have been able to guess, but with structure details, you can then make an informed decision how you would want to turn it into a biosensor. And last slide. The last reason for why we still want to look at lots of these different aptamers at a very atomic level is that there really isn't a lot of DNA aptamers binding small molecules in the database, like the PDB, for example. And I think without that, not only does it lack a lot of the information on binding mechanisms, but you have no resource, nothing to train anything to how to predict them better. And this is the example we actually, the structure I've just been showing you, we submitted it to CASP. It's usually for proteins. They predict proteins around the world. People rank the results, and you as a prediction team can be proud of your closeness or instead decide to work harder for next time. So we gave this one, and I don't know, I'll zoom in a little bit. Yeah, no one came even close to this structure. The very closest one was, I think, 11 angstrom, no, 10 angstroms RMSD, which sounds almost okay, but this is so small that there is actually nothing in common. There's no bases in common. Maybe there's some threes coming together. So there's a long way to go, which is great. It's a big field. But there we go. That's why we do it as well. So most of this was from Eric in my group, who's a lecturer, actually. And we also have great collaboration with both Philip and now Philippe. And thank you for your attention.